my gentle and very modern apes, and welcome to another episode of Science in Motion. Today we are looking at one primary paper, but we're also going to consider the abstract of another slightly earlier paper that I think allows for these two to kind of go in tandem with one another. So we're going to be discussing why humans are both the best ape at water conservation and why we are also the sweatiest ape. So this first paper, both of these papers are very recent, I give only the best to you guys, is titled Repeated Mutation of a Developmental Enhancer Contributed to Human Thermoregulatory Evolution. And I realize that might sound a bit intimidating, and to be quite honest, this paper can be a little bit of an intimidating read. When I read it through for the first time, I was like, okay, hold on, give me a sec. Let me go back and eyeball it over once more. And the methodology is still a little bit above me, not being a geneticist, so I'm going to go ahead and summarize that portion for you when we get there. But let's appreciate the paper that came prior to this one first, because like I said, they go in tandem, and that would be the evolution of water conservation in humans. This was from March 5th of this year, the other one from April 7th, I want to say. April 20th, even later, even more recent. Like I said, only the best. Now the abstract from this paper, does it give us the abstract or does it just give us a summary? It's from Current Biology, so that might be how they do things. The highlights: humans have evolved to use less water per day than other ape species. Our drinking regulation targets lower water dietary energy ratios. Our water conserving mechanisms are unclear, but they may include the external nose. And zoo apes may benefit from diets that match their dietary water energy ratios in the wild. So we'll go ahead and appreciate the summary of this one first. The spoiler, of course, is that humans are 30 to 50% more efficient at regulating our water, right? So to sustain life, humans and other terrestrial animals must maintain a tight balance of water gain and loss each day. However, the evolution of human water balance physiology is poorly understood due to the absence of comparative measures from other hominoids. So that would be your great and lesser apes. While humans drink daily to maintain water balance, rainforest-living apes typically obtain adequate water from their food and can go days or weeks without drinking. Here we compare isotope depletion measurements of water turnover in zoo and rainforest sanctuary-housed apes, chimps, bonobos, gorillas, and orangutans, with 5 diverse human populations, including hunter-gatherers, semi-arid savanna, and more. Across the entire sample, water turnover was strongly related to total energy expenditure, physical activity, and climate, as well as fat-free mass. In analysis controlling for these factors, water turnover was 30 to 50% lower in humans than other apes, despite humans' greater sweating capacity. Water turnover in zoo and sanctuary apes was similar to estimated turnover in wild populations, as was the ratio of water intake to dietary energy intake. However, zoo and sanctuary apes ingested a greater ratio of water to dry matter food, which might contribute to digestive problems in captivity. Compared to other apes, humans appear to target a lower ratio of water to energy intake. Water stress due to changes in climate, diet, and behavior apparently led to previously unknown water conservation adaptations in hominin physiology. So, why are we so good at saving water? The first and most obvious reason would be that we evolved on the open savanna. Our ancestors, due to the moving of the eastern African rift, came down from the trees and ended up being these silly, small-brained bipedal hominins wandering about until our larger brains came onto the scene in mid to late genus Homo. And that's pretty cool, because our bodies are adapted to that kind of living. We may not be as fast as many of the other animals, but we can run longer than the vast majority of animals on this planet, which makes us some of the greatest persistent hunters in the entire animal kingdom. We're outpaced by, I think 5 separate animals. Pronghorns can beat us, of course. Camels can beat us. Ostriches can beat us. Huskies can beat us. And I believe that might be it. It might only be 4. But we're very good at running for long periods of time. And part of that is because, 1, we can jog, and 2, we sweat. We are incredibly well adapted to cool ourselves off. And Sweating and water conservation go hand in hand, right? Living on the open savanna requires a very efficient use of water because we're not always having access to a fresh watering hole, nor are we getting a lot of the water from our diet, so we don't have the opportunity to eat these lush, juicy fruits like some of our cousins in the trees do. And of course, this means living in the hot sun has to make us efficient thermoregulators if we're going to be walking around all the time and not be fast. If we're going to go for long periods, we've got to be good sweaters. So this paper gave us the kind of support that we needed to confirm that yes, indeed, the first half is there. Because to be able to persistence hunt, you must be able to cool yourself off, and you need to not dehydrate yourself to death while doing it. So the fact that we're able to conserve water, that's step one. And step two, of course, would be, can we sweat to cool ourselves off and do so in a way that doesn't take all our water up? And the answer is yes. And the way that we do that is actually very interesting. So we're going to appreciate this second paper now. One of the most significant and distinctive physiological traits differentiating humans from the other primates is a reliance on sweating to cool off. Not just a reliance on it, but we're the best at it. We're the best sweaters. The effectiveness of human thermoregulatory sweating is underlain by the evolution of a drastically increased density of water-secreting eccrine sweat glands in human skin relative to that of other primates. So part of the reason why we're such good sweaters is because we are covered in sweat glands. We have 5 to 10 times the amount of sweat glands that the other primates, depending on the area of the body that you're looking at. We are just dripping water all the time. Here we show that the accumulation of human-specific mutations in a developmental enhancer collectively promoted the production of eccrine glands in humans by upregulating the expression of NGRELD1 transcription factor in the skin. This study reveals that a mechanism that contributed to the evolution of human signature thermoregulatory capabilities and underscores the importance of regulatory evolution in generating the modern human form. So the interesting thing about this particular mutation, the one that we're going to kind of discuss here, is that it's a mutation in regulatory genes. So it's a mutation in how our enhancers work, and we're going to be discussing this enhancer in a moment. But basically, you have this little enhancer. This enhancer acts on NGRELD1, which is the transcription factor that spurs your immature skin cells into saying, hey, instead of an immature skin cell, how about we be a sweat gland? And that's how we end up with a ton of sweat glands, which is really, really cool. So we're going to discuss how they did that now. Humans sweat to cool their bodies off and have by far the highest eccrine sweat gland density among the primates. Humans' high eccrine gland density has long been recognized as a hallmark evolutionary trait for humans, but its genetic basis has been unknown. In humans, the expression of NGRELD1 transcription factor correlates with the onset of eccrine gland formation. So that's what I was just mentioning. The NGRELD1 acts on immature skin cells and says, hey, be a sweat gland instead. In mice, regulation of ectodermal NGRELD1 expression is a major determinant of natural variation in the eccrine gland density between the strains, and increased NGRELD1 expression promotes the specification of more eccrine glands. Here we show that the regulation of NGRELD1 has evolved specifically on the human lineage to promote eccrine gland formation. Using comparative genomics and the validation of ectodermal enhancer activity in mice, we identified a human NGRELD1 skin enhancer, HECE18. This is our important enhancer here. We showed that multiple epistatically interacting derived substitutions in the human ECE18 enhancer increased its activity compared with non-human ape orthologs in cultured keratinocytes. Repression of HECE18 in human cultured keratinocytes specifically attenuated NGRELD1 expression, indicating this element positively regulates NGRELD1 in this context. In humanized enhancer knock-in mice, HECE18 increased developmental NGRELD1 expression in the skin to induce the formation of more eccrine glands. So when you pop this gene into mice, they sweat more, or rather they develop more sweat glands. Our study uncovers a genetic basis contributing to the evolution of one of the most singular human adaptations and implicates multiple interacting mutations in a single enhancer as the mechanism for human evolutionary change. So let's kind of get into this paper. Again, we're not going to go too in-depth into how they did it. I'll leave it in the description so you can read it for yourself if you want, but I'm afraid I'm going to butcher it if I get too much into it. As far as I understand, they scanned the genome, they looked for enhancers that are related to NGRELD1, checked that connection, and then double-checked it by implanting, knocking in that engrilled one, or not engrilled one, my mistake, the enhancer, the HECE18, into a mouse, and sure enough, that's what it does. So let's kind of read a bit more about it. Because of their furlessness, humans are often called the naked ape, but humans can also be referred to as the sweaty ape. Now, I like this sentence, but I don't know that I would call us furless compared to the other apes, because we, as some of you may know, we have the exact same number of hair follicles, like on average, as the other apes do, but our hair is comparatively quite thin. It's actually called vellus hair, and it's, helpful in a lot of ways. Again, it helps us with sweating. We don't overheat as easily. And as some PopSci articles argue, it actually helps us rather to rid ourselves of parasites a bit easier. We can see them and they don't have as good places to hide, and living on the savannah, there just wasn't that much of a selection to keep all that hair. But I digress. The evaporation of sweat is the major mechanism by which humans dissipate body heat, and humans have thermally induced sweat rates that are 4 to 10 times higher than those of chimpanzees, secreting 1 liter or more of sweat per hour. That is so much sweat. But yes, the way that we actually cool off is we sweat, so we produce water from our eccrine glands that end up on the surface of our skin, and that surface water evaporating is what actually cools us off. So it's sweating. It's not the act of sweating, it's the evaporation of the sweat. This dependence on sweating relies on 2 critical adaptations. The first is our loss of fur. I was just saying this. The first is our loss of fur, because fur reduces airflow over the skin. Human furlessness is thought to be an adaptation that enhances sweat evaporation. The second is a high density of eccrine sweat glands in the human skin. It is these glands that secrete the water humans vaporize for evaporative cooling, and it is their density that makes humans' copious sweat production possible. Indeed, eccrine sweat glands are the prominent appendages of human skin, whose densities exceed 200 glands per square centimeter in regions such as the face. In comparison, mean eccrine gland densities of macaques and chimps are 10 times lower. The importance of eccrine glands in humans is underscored by the risk of hypothermia in individuals with reduced numbers of these organs. Hyperthermia. Overheating, not cooling. How and when humans diverged from other primates to evolve their dramatically elaborated eccrine gland density is unknown. The evolution of humans' high eccrine gland density required modification to the program controlling the number of eccrine glands produced during development. Much of our understanding of this development program comes from studies of eccrine gland formation in mice. Mice retain the ancestral condition of having eccrine glands only in the volar skin of the paws. So this is similar to your dog. Like if your dog has kind of like wet paws, that usually they sweat through their paws. Most mammals do, the palms and the soles of the feet. And we kind of retain this. If you've ever had sweaty hands, we actually have a lot of eccrine glands in our palms and in our soles. That's why our socks stink. Here, where eccrine gland secretions regulate frictional contact with underlying surfaces. Here, eccrine glands are found in elevations of the volar skin, called foot pads and intervening interfoot pad surfaces of a mouse paw. Like hair follicles and mammary glands, eccrine glands are a type of ectodermal appendage and derive from the basal keratinocyte layer of the skin. So this is something that's kind of important too. Pretty much all of evolution, after you get the split of eukaryotes from prokaryotes, is going to be an adaptation of an existing structure. That's the name of the game. That's the case with limbs and digits, and it's also the case with many of your glands, from mammary glands to scales to feathers to fur to eccrine glands, for that matter. You're going to get basically a revamp of the classic skin cell, epithelial cells and things like that. In this case, we're talking about premature skin cells that are being spurred to develop instead into sweat glands. And it really is that simple. In contrast, we also know this because we can watch this happen in a lab, and sometimes we can get in there and spur immature skin cells into doing different things. There have been some really interesting studies, by the way, that have worked on scales and spurred them into becoming protofeathers, which has a lot of implications for the evolution of modern extant birds from theropod dinosaurs. In contrast, we and others have previously shown that the transcription factor, NGRELD1, plays a restricted role in specifically promoting the formation of eccrine glands in mice. NGRELD1 is expressed throughout the basal keratinocyte layer of developing mouse volar skin and specifically upregulated in eccrine gland placodes of the IFP. NGRELD1 knockout mice fail to form eccrine glands, and decreasing NGRELD1 levels results in specification of fewer eccrine glands in mouse volar skin. In fact, intrinsic differences of the cis regulation of NGREL1 are a primary cause of the fourfold greater abundance of eccrine glands in the IFP of FVB/N compared to other mouse strains. Strikingly, NGREL1 is upregulated in human fetal ectoderm coincident with the onset of eccrine gland specification. In light of these findings, we investigated whether evolutionary changes leading to increased ectodermal NGREL1 expression during development could be an underlying mechanism for the adaptive increase in human eccrine gland density. So, NGREL1 is what causes immature skin cells to become eccrine glands. What causes NGREL1 to act? Enhancer regions. So they were thinking to themselves, okay, can we find these enhancer regions? And of course they did. Identification of candidate NGREL1 enhancers in the skin. This is where they talk about kind of like what they did. I like this right here. There are no human-specific variants that distinguish the human NGREL1 coding sequence from that of other catarrhines, or Old World monkeys, a group of primates which include humans, apes, and Old World monkeys, so monkeys of Asia and Africa. And most evolutionarily relevant changes are likely to reside in regulatory DNA. We therefore first investigated whether humans have altered NGREL1 regulation by screening for enhancers with potential to control the locus in eccrine gland development. So that's kind of what they were looking for. Then, that's a cool chart, by the way, you should check that out if you're interested. Then they discussed the enhanced activity of the ortholog of ECE18. So among the 5 that they identified of these enhancers, they went with 18 because it overlapped with most of the epigenomic signatures that are successive to enhancer activity from other published data sets. So they talk about doing that for a little bit and why they picked that one and how they influenced it. Multiple mutations underlie increased ECE18 activity during human evolution. So because ECE18 activity is higher in humans, we reason that human-specific sequences might underlie the potency of this enhancer in our species. To investigate the specific changes of the higher activity of the human enhancer ortholog, we first tested this genomic fragment that contains the highest number of derived human substitutions. So they find that only a handful of low-frequency variants were detected, and essentially, they found that replacing even a portion of the genomic interval spanned this particular enhancer with orthologous chimp sequences was sufficient to reduce the activity of the enhancer in luciferase assays, indicating that the importance of human sequence changes with this particular enhancer. So essentially what they did is they reverted some of them back to chimp levels, and they got reduced expression, or enhancement, I suppose, of NGREL1. So you turn it off, you reduce it to the chimp levels, and you get less sweating. You get less sweat glands, which is, kind of all she wrote. They talked about a conserved mechanism, which is kind of cool. Overall in silico analysis predicted anthropoids, which is going to be like your Old World monkeys, to have the highest affinity SP1 and SP1A, and SP1B motifs among mammals, which is interesting. I'm sorry, anthropoids, simiiforms. I should have been more clear about that. That's not Old World monkeys. Old World monkeys is catarrhines. Anthropoids includes catarrhines and platyrhines. It's late, so forgive me. Then they talk about how ECE18 modulates endogenous NGREL1 expression in human keratinocytes. So they basically talk about kind of how that works and what it does. I apologize for not getting more in depth with this, but again, it's a bit over my head, and I don't know that everyone would find it super interesting to get in depth with it. And I'm also going to be more prone to making mistakes if we get kind of in depth to the degree where we're reading every paragraph. So they talk about how it upregulates NGREL1. We've talked about this. This is the art of writing a paper, finding different ways to say the same thing. So the discussion. Natural selection has shaped many unique human attributes, but the genetic bases of these features are largely unknown. The findings of this study implicate a genetic and developmental mechanism harnessed in evolution of one of the most defining physiological adaptations of humans. That human ECE18 not only positively regulates NGREL1 expression in cultured human skin cells, but also in the developmental model of eccrine formation, provides compelling evidence for similar functionality for this enhancer in humans. Combined with the finding that human-specific mutations have increased the relative strength of HECE18, our study supports an evolutionary scenario in which a stronger HECE18 produced upregulation of ectodermal NGREL1 during development, and a concomitant increase in the number of eccrine glands specified in human skin relative to that of other apes. So basically, mutation in the enhancer makes more eccrine glands means more sweating. And it's really as simple as that. And one thing I forgot to mention in that methodology and in their results section is that what they found is the mutation that actually acted on this enhancer was multiple successive mutations on the same region. That's it. Just repeats. Same mutation over and over again. Not that complicated, as very little of evolution tends to be. Again, most of the time it's adapting a previous structure. An intriguing feature of HECE18 is the accumulation of multiple point mutations. I was just saying that. Scattered throughout the enhancer have collectively increased this element's activity in keratinocytes. Derived human base pair substitutions at conserved primate SP1 binding sites appear to have played the greatest individual role in increasing HECE18 activity. Intriguingly, 5 of the remaining derived substitutions which contribute to increased enhancer activity lie within the human accelerated region, which is this one right here, 2XHAR20. While future additional work will be required to tease apart the epistatic interactions between mutations within and outside the HAR and to unravel the specific steps of the HAR playing in evolution of higher eccrine gland density, it is worth noting that our findings directly implicate bioinformatically defined HAR evolution of an adaptive human trait, which is really, really interesting. So then they discussed how it evolved in a stepwise manner. This is something that they again tracked in their methodology, and suggest that the element could have contributed to the evolutionary increases in the density of Homo sapiens at multiple points during human evolution. So it's not an all-at-once thing. Our study raised the possibility of sustained selection on multiple functional derived human mutations, which together account for the increased activity of this enhancer relative to other primates. To understand this, in which part they're talking about how they need to understand the variants a bit more. And then they said, as a final point to consider, whether the evolution of HECE18 can explain itself, the human's tenfold increase in eccrine gland density over that of other apes. They noted that HECE18 produced a subtle increase in eccrine gland number in the engrilled-1 sensitized mouse knock-ins. In light of finding that it is not an essential part of endogenous regulatory machinery in controlling engrilled-1 levels in the mouse, the mild effects of introducing the human enhancer may be reflective of an attenuated functionality of HECE18 in mouse skin. Determining whether there are associations between present-day HECE18 polymorphisms and variation in the human eccrine gland density will help shed light on the relative importance of this enhancer to the specification of eccrine gland density in its endogenous human context. Further studies to comprehensively map the enhancers that control ectodermal engrilled-1 levels during eccrine gland specification and functional follow-up of the significance of derived human mutations therein may implicate additional engrilled-1 dependent mechanisms that may have played a role in human evolution of eccrine gland density. And then they talk about some of the other candidates for enhancers. So they're basically like, this enhancer is doing a lot. Maybe it doesn't do it all. Maybe we need some of these other very close by enhancers that probably would mutate in a very similar way. So let's see here. Let's get to our conclusion. Our conclusion is just going to be here. The mechanistic insight into how HECE18 is regulated provides a foundation to interrogate these pathways and to investigate the origins of other related traits, such as the generalization of eccrine glands to non-volar skin. Moreover, because of the importance of engrilled-1 in the development of other organs and tissues, including derived human brain structures such as the cerebellum, our findings may shed light on differences in the modulation of human engrilled-1 in these contexts and the etiology of other evolutionary significant human phenotypes. So, it's as simple as this. When you're trying to get a persistence hunter, what you need is a good water conserver and a good sweater, or not necessarily sweater, a good thermoregulator. In the case of us, it's going to be sweating. So high water conservation perhaps came first, the ability to conserve water when foodstuffs that are high in hydration are kind of absent. And that ability would have kind of paved the way for this ability to kind of waste water sweating, right? You don't have to use it all for staying hydrated. And in the case of sweating, all it was subsequent mutations in this enhancer region that then in development prompts engrilled-1 to spur the development of new eccrine glands, more eccrine glands from not fully developed skin cells. And that's it. Which is really cool. These two papers kind of in tandem with one another is pretty dang neat. What I would like to see is kind of a look into the genetic minutia, I suppose you would say, of how we actually got this water conservation in humans. And they might go into it more in the paper. I kind of need to, I've read it, but it's been a month or so. And if memory serves, I think they did get into it a bit, but not nearly as much as this one where they actually went down to the genes, to the enhancers, which is quite neat. So, I hope you enjoyed this episode of Science in Motion, and I hope that all of you gentle and modern apes take care of yourselves and join me next time. Stay sweaty.